I don't have my gavel, but I'd like to call the 1255th regular business meeting of the Entomological Society of Washington to order. As most of you know, the museum is currently closed due to the lapse in government funding. So we are meeting virtually this evening. Thank you all for joining us on Zoom. Uh, I'd like to start asking Secretary Gary Havel. Secretary, will you please read the minutes from the October meeting?
Yes, thank you very much. The 1254th regular meeting of the Entomological Society of Washington convened at 7:00 p.m. on the 2nd of October 2025 in room WG 33 of the National Museum of Natural History. President Teletus Samoz welcomed the 14 members, 11 guests, and 15 virtual attendees. Recording Secretary Gary Havel read the minutes of both the May meeting and the annual banquet, followed by approval from the attending members. Membership and Communications Officer Matt Buffington happily announced the recent membership request of the following 17 applicants: Dana Brennan, Catherine Malinsky, Sydney Bennett, Caitlin Bloomer, Awanti Shastri, Jennifer McHenry, Jackie Chitty, Emily Vasile, Alejandro Estrada, J. William Stubblefield, Diana Percy, Arnold Mackey, Courtney Zenz, Tim McCabe, Catherine Turk, Beatrice Rodriguez, and Zeno Mangual. Past President Don Weber announced that present officers have agreed to continue their duties during next year. Alan Norbaum has agreed to running for president of the society. A slate of officers for 2026 will be presented during the November meeting. Members are encouraged to nominate themselves or others for the positions. Don Weber displayed new entomological books for examination. Don also announced the annual free lecture at AAAS on November 20th, featuring bee expert Chris Granger. Elmore Brown was asked to speak about YEG, Young Entomologist Group, and presented an informative and loquacious outline of activities of this organization. With a series of images, Elmore presented the purpose, organization, and activities of YEG, saluting the leadership of David Adamski. YEG was established in 2012 as an outreach organization of the Entomological Society of Washington. Youngsters include those of ages 5 to early teens. Among Smithsonian participants in YEG programs have been Dana Roach and Vishai Malakul. Program Chair Alan Norbaum introduced the speaker of the evening, Merrick L. Sundquist from Arlington, Virginia, who is 14 years old and the youngest person to ever speak during a society meeting. The title of the presentation was The Effect of Substrate Grain Size on Antlion Larvae Funnel Building Efficiency. During YEG trips, Merrick noticed antlion funnels and decided to investigate these fascinating insects. Antlions are known to mostly prey on ants and have 3-year lifespans. For pupation, they make a cocoon of sand.
I think so.
Silk.
Because that's not the guy's name.
There are some 2,000 worldwide species. Antlions of the past are known from 46 million years ago. Merrick mostly analyzed antlion larvae by providing soils of different grain sizes. Larvae were found to favor clay sand for development. Videos of antlion larvae in their funnels were shown, demonstrating their ability to remove ant carcasses by flipping them out of tunnels with their heads. Merrick plans to continue his research with antlions, with some attention to the exact sizes of females. The meeting was adjourned at 8:05 p.m.
One possible correction, Gary.
Yeah.
I think Chimo should be recognized as a new life member
Okay,
society, not a new member.
okay,
Thank you.
sure, thank you.
I'm sorry. Any other addition or corrections to the minutes? Heard nothing, so can I get a motion to approve the minutes as read?
So moved.
Seconded.
Second, thank you very much. The minutes are approved. So let's move to officer reports. Please, officers, share any relevant updates.
Hi, this is Matt Buffington. I'm the Membership and Communications Secretary. And I'm pleased to read in 3 new members to the Entomological Society of Washington. Arata Baba of Bainbridge Island, Washington, Marion Bundens of Knoxville, Maryland, and John Schneider of Houston, Texas. Welcome all 3 to the Entomological Society of Washington.
Thank you and welcome. Webmaster Lourdes Chamorro, do you have any update?
Just a small one. We have a couple of things set up to debut in the next month. One is a merchandise store that we're still trying out some of the glitches. We'll offer mugs and cups and shirts and things like that. We're trying to just do it at cost, more or less. But we want kind of have people maybe give these as gifts if they like and have the logo. And if you have ideas for other items or drawings to add to, we can actually customize anything. And the other is a campaign around the time of Thanksgiving. We're thinking of having a Giving Tuesday. Kind of there's this Black Friday and then Cyber Monday and then Giving Tuesday on the Tuesday after Thanksgiving. So kind of keep an eye out for that. And we'll be doing a little campaign for that. That's it.
Thank you very much, Dr. Chamorro. These are 2 updates that I'm especially very excited about. Do we have past President Don Weber?
No,
He's running a little late. I'm texting him now.
Okay. So maybe when he joins us, I believe he's gonna have a report as well about the upcoming elections. In the meantime, let's move to the introduction of new members with us here today. Is there anybody that is a new member or a visitor joining us today for the first time who would like to introduce themselves?
Yeah, well, hello. Sorry about that. Take 2. I'm Ben. Good to see everybody. I'm especially glad to see some familiar faces today, considering the shutdown. I haven't officially joined ESW. This is my first meeting, but at some point I'll get around to officially joining ESW. So thanks for having me.
Thank you, Ben, and welcome. All the meetings are open to all, so you can keep coming to the meetings on Zoom or in person without being a member, but of course we would love to have you as a member. Anyone else?
We have a few on the chat.
Oh, okay.
Pauline Baylor from Virginia.
If I put the chat up here. And yeah, so welcome, Colleen. We are also very excited to have you here. Anyone else? Okay.
I would like to just take a moment here and remind anyone who is a new member or considering a membership. With membership comes privileges, and that means you get to publish in the infamous Proceedings of the Entomological Society of Washington for free. up to 2 color plates. You pay additional for color plates beyond 2. And starting next year, open access, free open access for all publications. That's one darn good deal. So please keep us in mind and join the society, reap the benefits.
Yes, thank you. We also have 2 people from Michigan, William Ford and Julie Kravis. Welcome. So let's move to unfinished business. Is there any unfinished business from the previous meeting that we would like to address tonight? What about new business? Is there anything that anyone would like to bring to the floor?
Talita, Don just joined.
Okay.
Yeah, thanks.
So
I do have an item. Can I share my screen or do I wait, Talita?
I'm not the host, so I think Alan can let you share your screen.
Oh, I am screen sharing.
Yes, you are. Go ahead.
Okay. Well, as you know, next month we have our annual election of officers. And in addition to the slate listed here, there is the opportunity to self-nominate or nominate anytime up to and including that meeting. I think we have a nice slate and everyone has their statements there. And I think, Lourdes, were you going to post that on the website or send it to the members, or how are you going to do it?
Yes, I'll send that to the membership after this meeting, so people can become familiar with the candidates and then be set for the vote. Thank you.
And decide if they don't like them, they can run themselves, right?
Right.
So, yeah, Matt, I think I missed yours because it was coming from USDA to my USDA. Sorry about that.
I figured that. I quickly generated one up though, but yeah.
Yeah. Thank you. So you can see the slate there. And I don't think I have to say anything else. Other than that should be publicized, and then we'll formally vote on it at our, I guess it's considered our annual meeting next month.
Yes, thank you, Dr. Weber. And as a reminder for everybody, the next meeting, our annual meeting, is going to be a very important meeting. So I would like to remind everybody to be with us in the first Thursday of December, hopefully at the museum.
Should I unshare that? I guess.
Yes.
Otherwise,
And
we won't get a presentation. Everyone would get it then, I guess, Lourdes, right? Or
They will, yes. I'll
okay,
send
great.
it,
Great.
and then the membership will get it through
Super. Okay.
email. Yep. Thank you.
Okay, so let's move to the presentation of notes and exhibits. Even though we are meeting virtually, if anyone has anything to share, please go ahead. Matt, do you have something?
There we go. So, boy, do I have some notes and exhibits to share. A friend of ours out in California, Dr. Lynn Lebeck, a graduate from Riverside back in the day, has a large book collection that she's been sharing with me and Lourdes. And she shared a few volumes that are of particular interest. One is the Beetle Collector's Handbook, which is a very old volume from the UK, I believe in the late 1800s, Nope, 1902. Just a gorgeous book on all beetles and how to collect them, everything that was known in 1902. That one was for Lourdes. But for me, were the collected notes of C.V. Riley. 2 volumes here of all of his literature. Someone bound them all into a kind of library
Oh
volume.
my gosh.
So it's just this remarkable. It needs to be rebound. Dawn, I was really excited to show this to you because
Oh yeah.
I mean there might be some actual handwriting in here. I'm not really sure. There's
Oh yeah.
there's some pretty neat stuff here. The
Sure.
other volume is right here, but it's very broken. The spine is unfortunately split, and it's been kind of
So
taped
these
together here.
were these handwritten notes or pamphlets that he sent out or what?
No, these are like his it looks like collected reprints, but I think
Okay.
there might be handwritten notes in it. I need to kind of go
Oh,
page by page.
how did she come by those?
I can't remember. She collects old. Yeah, she's in biocontrol out there in
Yeah.
yeah, you know her, in Clovis, and she just collected these. These were discharged from or withdrawn from Bowdoin College Library.
Bowdoin.
Bowdoin. Is that Bowdoin? Is that how it's
That's in Maine. Yep.
okay? So yeah, pretty amazing stuff here. I was really excited to share these with the society that he helped found. And I have to note he had a presidential address, which is reprinted in volume 1, issue 1 of the proceedings, and it is 11 pages long. So I think, Talita, it's up to you to reinvigorate the presidential address. And so we're looking forward to something lengthy on of entomological interest to you.
That sounds very nice. It would be great if you could bring it to December meeting because I really want to put my eyes on it.
Yep, I'll bring all 3, in fact.
Thank you very much, Dr. Buffington. And don't unmute yourself yet, because if we don't, do you have something to
I
share?
am unmuted and I want to show you another very British book that just came out. It's called how is that not working? I gotta hide behind it. Can you see it now? Can you hear me now? An insect a day. I'm gonna try and use this in the coming year. Every single day has a color picture. A few days ago was the St. Helena giant earwig, the world's largest earwig. And the Wallace's Sphinx moth is coming up. The proboscis of this moth is up to 30 centimeters long, and so on. It's charming. Oh, gosh, it's not showing, is it? What is happening? I don't know.
It's because you have a background.
It's because I have a background. So
yes.
It happens now that we all work for little machines. And if we don't do what they say, we're gonna, okay, now maybe it works better. Now it blurred that out, see? Okay, so what should I do? Adjust the background and effects. Okay, and we're gonna do none. So there it is. Okay.
Yes.
Anyway, it's a cool book. And it's put out by Batsford, which is very British. An insect a day. I recommend it.
It looks amazing.
Just kind of keeps you buggy every single day.
Yes, if you could also bring it to the December meeting. So I normally like to see with my fingers.
Oh my gosh. Okay, I got to mention one other day. 8th of October is Halobates. Isn't that our society insect? No. Is it? Jared? Or no? What is our society insect? Come on, Matt. Stop making faces. What is it?
Some sort of Jared.
It's not a Jared. It's the smaller, whatever.
Yes,
Okay, Warren knows. Warren's gonna say it.
it is a water bug. It's named Rhumatobates.
Oh,
I feel like one right now.
oh gosh. Okay, sorry about that. I'm lowering my hand.
I think it was named by C.B. Riley. I could be wrong.
Rhumatobates. Oh, that would make sense, wouldn't it?
And the type locality is Plumbers Island, Maryland, in the Potomac.
Oh my gosh.
You have to check me. Don't publish that until you check.
Not publishing it.
That would be incredible. That would be so cool.
I think there's actually a little article on the cover and the logo, and I think we need to kind of publicize that. And I was going to put it on the website,
There's
so
an article.
I'll get that. I'll get on that.
Thank you. Anything else that you would like to that anybody would like to show?
I have something to show.
Mm-hmm.
All right, give me 1 second. Hello.
Oh,
So I, for the last year, have been collecting with a Malaise trap in my backyard, and I did an entire year of collection from one spot in my parents' backyard, and I just wanted to share all of the samples that are nice and ready to go. So I just cracked through the first one of them this afternoon and did all the Heims because I like the Heims.
Take
And
the weevils.
we'll get there. And on the week ending in June 2nd, 2025, we collected 30 families of Hymenoptera in my backyard. I just think it's very exciting, and I wanted to share with some people who I thought might care a little bit. So very cool. Thank you.
Yeah.
that's very nice. Keep them refrigerated if you want to do metaphor coding in the future.
I'll use my big -80 in my parents' basement.
Just move the turkey off to the side, you know.
Yeah.
Thank you, Luke. Anything else? Okay, so we're gonna move to our announced topic of the night. As you probably saw in the email, the scheduled speaker is unable to present due to the lapse in the government funding. So instead, we will be presenting a pre-recorded lecture. Dr. Buffington, can you give us more information about what we're going to be watching?
I sure will. Thanks everyone for joining us tonight. When we had this bit of a crisis on our hands, I quickly reached out to a few people that might be able to give talks. Of course, ESA and ECN start like tomorrow in Portland. So a lot of people were mobilizing, a lot of people were occupied. But I contacted my former advisor and very good friend, John Heraty at UC Riverside, and he explained he had just given a talk to the Essig Museum in Berkeley, and they had a recording of it. Could we just use that recording? And that's exactly what we're going to do. So we have it with his approval, if you will, and he is driving right now to Corvallis, and we're hoping at the end of this, he will be in a position of stability such that he can do Q&A. So assuming he remembers what he talked about in his video. So we'll have to make that leap. So for those of you who don't know John Heraty, he's got his undergraduate at the University of Guelph and went on to with a degree in biology and some systematics. And he went on to do his PhD at Texas A&M University with Jim Woolley on Eucharitid wasps, the chalcids that we're going to hear about tonight. From there, he went to Carleton and then to Smithsonian on back-to-back postdocs. And then in 1995, got a faculty position at UC Riverside. And I met him, it seems like about 1 year after he became faculty, and he's been a part of my personal research life and family ever since. So it's a real honor to have him give this talk, even though it's kind of remotely. But we'll get to talk to him at the end. So stay tuned, and Lourdes is going to fire up the film for us.
I'm dealing with. And so, as I say, it's about chalcids, eucaridids, and basically trying to look at that integration of systematics and biology. So hopefully it's not going to be, hopefully I've got enough time to get through all of this. Oh, there we are. Okay, so this is me. Long time ago in Brazil, after I just got my assistant professorship here, I'm standing out in a Brazilian jungle, sweating badly and stinking very badly, barely able to stand myself. In the middle, I'm in New Zealand going after a rare little insect down there. And on the right was just last September, still trying to go after an ant host of a particular Eucharitid wasp that I'm after. And that little insect photo is a Eucharitid. My studies over the years have taken me around the world, all those little red dot places, in search of just basically looking for eucaridids and hosts and biology and everything else that I can do with them. I want to sort of reflect a little bit on my background. I am a first generation student. My parents were basically, my father was in construction supply and my mother was a bookkeeper. But they had great aspirations for me, and they said they just wanted me to get a higher level degree. Of course, my mother wanted me to be a doctor, of course. So I went off to the University of Western Ontario, had a very bad first year. I got a letter from the university that said, we strongly suggest you reconsider whether you should return to the university. So I did not. I went to work in a cement factory for a while, decided that wasn't good. And that's when I ended up at the University of Guelph. And I met this wonderful person, D.H. Pengelly, who was basically a really good mentor there. And he introduced me to the world of chalcids through these wonderful little study boxes. I was also hanging out with a bunch of undergraduates. They looked a lot younger back then. And all of us, these people were, really enthusiastic about collecting and getting out in the field and doing things. So that really sort of provided an inspiration. I then worked for 10 years in biological control, where I worked on leaf miner pests in apple as one of the good things I was focused on. And I started looking at parasitoids. And of course, the parasitoids started sucking in my scientific interest. Then I met Lubomir Masner at the Canadian National Collection, and he introduced me to this wasp and said, you should work on these. And that was it. I had these focus for the rest of my life. Also, during that time, I had the pleasure of hanging out with a couple of times with Bill Brown from Cornell University. He worked on ants. And he would extol great stories about all his collecting adventures around the world and things like that while we camped out in his backyard and things like that. So that sort of, helped to sort of inspire me again to go on these worldwide adventures to look for what I'm after. And then I ended up at Jim Moley at Texas A&M University. He was basically doing cutting edge phylogenetics at the time using these wonderful little computing systems like a high-tech VAX computer that he had in his laboratory, but that was basically also sort of integrated with the transformation of what I was doing. So anyway, back to the actual topic of what my talk is. So what are Chalcidoidea anyway? Hymenoptera, a broad group, we all recognize what are bees, ants, and wasps. They're easy to recognize. Chalcids, these ones on a dime, we can look at those and say, okay, here's one sample from Peru. We get 7 different families on there. And they get down to those ones like that little tiny one where it says, in wasp we trust on that dime. You can't do the same thing with mammals. You'll never get, there's a raccoon in a trap, but you're never going to get all of those into a single trap. So it's kind of, just the diversity of things we're looking at is tremendous. Chalcidoidea now have a brand new 50 extant families and a whole bunch of subfamilies and genera and species. We have these high estimates because basically they're attacking and killing a lot of things. So we have, a long way to go to try and get an idea of what these things are. But slide sort of gives you an idea of the morphological diversity. They're extremely small. We can get down to things like megaphragma, and this is a to-scale image with a paramecium and amoeba to get you that size relationship. This is the smallest living terrestrial insect in the world, and basically this is actually a male mimerid, and it's you look at that thing and you say, okay, well, it's got we think these are the antennae up in here. That's probably the mouth. We know it's a male because it's drooling. Actually, it's got genitalia too. And if you look at the legs, there are no tarsi. This is just the aurolium with the tarsal claws on there. And, you know, how would you ever make an association about this particular group? Well, this all came down to a fellow named Mockford, who was working on a culture of sosids in Costa Rica. And he came up with this little male over here of Dicopomorpha associated with a female Maimerid over here. So it's probably using those big aurolia to maneuver itself either in the egg to mate with the female, or it uses it maybe even to ride her when she leaves the eggs. We really don't know that much about it. Chalcids are diverse. They're primary, secondary parasitoids, encoins, gallfarmers, pollinators. They're endoparasitic, ectoparasitic, whatever you can think of. They attack everything from nematodes to mites all the way to other insects and things like that we know them best for. Because they're going after things that are in our agricultural systems, because they're after things in our agricultural systems, we care about them. So they do leave for a big focus of interest. And one of the reasons we have the University of California, Riverside down here was the beginnings of citrus and basically looking at parasitic wasps. We're trying to control some of the pests there. This is John Noyes in the British Museum. I have no idea what he's doing with that tree, but he decided he wanted to find out what he could get in 6 hours continuous sweeping in forests in Costa Rica. And so at one location, he can come up with almost 10,000 or 10,000 individuals and 787 species. And I would say that those are morphological species. They're not even dealing with the high number of cryptic species that we might expect in things like Aphelinidae and Mimeridae and some other different groups. Yoshimoto, back way in 1975, had an idea of phylogenetic relationships in the group. They're kind of odd, but we are seeing things like Mimerids coming out as basically sister group to something of this little mess down in here. And we got trichogrammatids up in here and some groups that we can sort of recognize. But most of these are sort of showing up in the Eocene Baltic amber deposits and things like that. So we have an old history that dates back to the Jurassic for the entire group. We sort of come up with this little idea that we think we have Mimerids as egg parasites, trichogrammatids as egg parasites, and Eulophids as other egg parasitoids, and then we get these others. But we really, at this point in time, didn't have much of an idea of whether that was sort of true or not. So Gary Gibson, 1986, comes up with a morphological phylogeny. He introduces a new group here, the Mimeromatids, which are these cool little creatures here, really, really tiny. They have little accordion heads that I don't know what they use them for. And then we have Mimerids up here, which is sister group to the rest of the Chalcidoids. But you can see that nice little question mark here about, how we're dealing with this. So this was the beginning of sort of real relationships with character-based. So where are we now? This is a quick sort of jump through. In 2011, we had 720 taxa using what I call the cheap genes now to try and get an idea. We didn't have much backbone support. We then used morphology to come up and look at the whole group to try and get a better idea. We got some better groups coming together that we didn't have before, but there were still questions about where this was going. And then I'm going to use this red box here to focus in on something that we'll be talking about later, which is the perilampids and the Eucharitids, to sort of give you an idea of where they're placed in this classification. So then Ralph Peters down there in the lower left-hand corner basically came up with a transcriptome analysis of the Chalcidoidea. And, this was looking, there's my Eucharitids and perilampids there. We're looking at something where we have, a huge number of amino acids, 37 Chalcidoidea, which isn't much. 11 out groups. We get poor backbone support. So in the green up in here, there's not much support, but we're getting good deep level support down here. And you can see we got Eulophids, trichogrammatids, and Mimerids. So we got this, again, the support for Mimerids, trichogrammatids, and Eulophids. Why do I care about this? Because egg parasitism may be the bottleneck that Chalcidoids had to get through to form all the diverse morphology that they have now. So they had to be super small before they became anything bigger. Introduction. So this is the egg parasitoids, which you saw in that introductory slide, the Mimerids, and they're going after eggs and plants. We have trichogrammatids going after a variety of things like eggs and things like that. And then we have the Eulophids, which are, sort of rarely egg parasitoids, but they're mostly larval parasitoids within those different things. So, sorry. We decided to revisit the transcriptome analysis, and Junja Sang was my postdoc at the time, and she worked with adding more taxa. She worked on adding a new ortholog set for Chalcidoids that we've been working on, and there's a bunch of data there that you can always go back and zoom and get an idea of what all those different things are. She came up with a different analysis, and sort of the results were pretty much the same as what we saw before, except for one thing. If we looked at the concatenated results, where you're lining up the genes one end on other and analyzing all in one analysis, we get mimerids, then trichogrammatids, then eulophids, which is the same as what the Peters paper said. But if we looked at the astral results, which is looking at, the summarizing all of the individual gene trees, we got a different results with mimerids and then trichogrammatids more highly nested and eulophids in there. So we weren't getting quite that result that would suggest egg parasitism was definitive. So this is T1 and T2 hypothesis for reference. The gene conflict we're looking at within those. So for the concatenated data for the T1, we got mimerids and trichogrammatids sort of ladderizing there. For the T2, we get this conflict about their nested trichogrammatids are nested up higher in the tree. So we thought, okay, so do the genes individually support something different? Well, it was pretty messy. So this is the gene-wise log likelihood score. We have the T2 hypothesis and the T1 hypothesis. And I think you can get an immediate idea that we have a lot of genes that are just conflicting with each other on almost an equal basis over those two different hypotheses. So this is not good. It's not sort of lending you to an idea that you're definitive on what the actual answer is. Okay, does adding more taxa help? But first I need to introduce you to two new players in the system that were not in those transcriptome analyses. Myromatoidea, this is the one that I mentioned that Gary Gibson brought in. It's an egg parasite is basically up until 2022, we had no idea what this thing did. We had an idea that it attacked sosids, but in this brilliant paper that I think deserves a Nobel Prize, David Hunsberger was able to actually demonstrate that they were coming from the eggs of sosids in Hawaii. So we had a definitive egg parasitism record for this. Appreciate that. The other ones are the biomorphids, which are basically considered to be the sister group of the rest of the chalcidoids after mimerids. We have two genera, Rhodowida and Chiloe, and it's amazing to think that these things were only just described in 1987 and 2000. So an entirely new family and two genera, and notice there are ones from New Zealand and ones from Chile. We have a past distribution. They're well known in amber, and they're mostly, getting up into the Palearctic region and Canadian amber. So we have a good idea based on fossils what they are, but they're all fossils. And the only extant distributions we have are this Rhodowida, which is found in New Zealand, and Chiloe, which is found in the southern end of Argentina. So for dating purposes, these become a really important thing for our phylogeny to be able to get these. So we go to our team. There's Ralph Peters, Austin Baker, my student, Lars Krugman was from Stuttgart University, and of course, Mr. Grumpy Jim Woolley here. And we basically we're in both New Zealand and Chile. This is Chrissy Dominguez down here, and we were going around looking for, trying to find out what we could come up with. And it turns out that rhodowitids in general are associated with moss habitats. And we're pretty much certain that the reason they're associated with this is because they're attacking these wonderful little pellurideids down in here. We have not got a definitive egg relationship going on here, but if you look at the ovarian egg of the pellurodeid, it's basically about the same size and shape as that Chiloe. The Chiloe is really cool because you get it in a plant trap, you transfer it to alcohol, you're sorting it in your dish, you see one, you pull it out, you put it on a piece of paper, and it starts walking. There's something about that plastron on there that keeps anything away. So we had a great system where we could have looked at active host, whether it would go after an egg, but we just couldn't figure out where that pellurodeid laid its eggs to be able to test it. So if anybody wants another project to do, got a great place for you. Go in Chile to look for these things and figure out their biology. Okay, more taxa, different approaches, looking at anchored hybrid enrichment now, evolution of molecular methods, and ultra-conserved elements. We had the wonderful pleasure of, this is Junja Zhang and Roger Burks, which were working in my lab at the time, and we had Jean-Yves Rasplus and a street crew in France. We were looking at anchored hybrid enrichment methods, and they were looking at ultra-conserved elements. And we had these two large little data sets, and we thought, and both of them are including myromatids and biomorphids, so we were sharing taxa and getting things back together. And what do we do with that? It's time for a merging of the data sets. So we combined the data set and we came up with our 433 taxa and a bunch of, basically a lot of genetic information on here. And that's our phylogeny that just came out in 2024. Now, again, we come up with an nice little egg parasitic basal on here. So we got mimerids, myromatids, and biomorphids. That biomorphids, again, being that question mark, but we're pretty sure that they're going to be egg parasitoids. So again, it's really that emphasis that chalcidoids had to reinvent themselves by coming through this little small egg bottleneck. And that's why they look so different. Anyway, now we have 50 extant families, 13 Entomological, which we still can't figure out where they go because they bounce around on the phylogeny. And we have 3 extinct families, and all of this was basically part of a huge collaborative effort. And we made our decisions on family level relationships at a workshop in Prague in 2018 with a number of people from around the world. So it was a collaborative thing. And then if you want to know anything more about these things, we now have Chalcidoid Wasps, new book, and I can tell that everybody is absolutely thoroughly excited about this book. So please get out there and get your copy now while they still last. Okay, so that's enough about chalcids. Now we're going to talk about Eucharitids and back to this group, which are where they're located on that phylogeny. Eucharitids belong to what we have now defined, and every analysis comes up with what's called a planidial larval clade. And it's basically everything that is defined by having this nice little planidial larva, which is an active larva that is basically, this is the first instar, and it's got to make it's basically responsible for making the host association a little more so than what the female is, however she's laying her eggs and doing things. So it's an extremely diverse clade, but it's coming out monophyletic in different analyses. We also had an anchored hybrid enrichment analysis of the planidial clade. I'm not going to go too much into this. There's a bunch of data on the side there that sort of defines it. But again, we seem to get these same relationships of Eutrichosomatids, chrysolampids, perilampids, and Eucharidids. And we're even getting sporangia, which may actually have like a planidial style larva based on what I've been reading about their behavior. So morphologically, we have this Eutrichosomatidae, Jambia, which is in Chalcidoid, Chrysolampidae, Perilampidae, and Eucharididae with a number of different subfamilies. Now you look at those individuals on the right, and there's no way that you would ever necessarily believe that they all belong to the same group of wasps. There's no morphological, there's very little morphological evidence that would come across and support them. Molecularly, they do come through very nicely. They're all united by the possession of a planidium. Now you might say a planidium is, this one is what you recognize. This is a perilampid planidium over here. This is a eucharidid planidium within an egg over here. This is one standing on end. This is one feeding on its host over here. And this is the one that's the Eutrichosomatid. It was the Eutrichosomatid. We didn't know anything about that one particular group. And we had to do sort of a search to find out exactly where it was found. Sorry, need to go back. It turns out that this is found in the seed pods of acacia. The wasps lay their egg in among the eggs of the weevils, and basically as the weevils develop, the wasps are sort of tracking them by having these little soft body planidial larvae. This is to give you an idea of what the planidia look like. These are plants on Mercine collected at the Waikecha Peru station at 2,700 meters, where we've got Eucharidids laying their eggs on the plants. Those planidia are all located around extrafloral nectaries on the plants. Their host was a Phaedola that was nesting at the base of the plants, but we never were able to sort of figure out, we never found any ants up in the plants to figure out how they were getting from the plants down into the actual ant nests. Eucharididae, the ant parasitoids. So we're going to focus now just strictly on those over some of these other groups. Okay, this is based on, there's a tremendous history behind this group based on sort of the legends of the insect world. So William Morton Wheeler was an ant specialist, and he provided these first detailed drawings of and information on the biology of Eucharidids from Texas and Colorado. Absolutely meticulous work, but he never thought Eucharidids ever left the nest. He always thought they were just within the nest at all times and just ant parasitoids within there and never leaving to lay eggs on plants or anything like that. So William Morton Wheeler was a start. Curtis Clausen, who was here at Riverside, actually looked at comparative morphology and behavior of Eucharidids. He looked at later instar larvae and how they oviposited and summarized a bunch of biological information, which formed a really strong foundation for the things I started looking at. So life history. So Eucharidids come out, they lay their eggs on plants, they have their eggs, they can lay them into flower buds, into fruit, such as this is just a small little fruit with massive eggs in here. They can lay their eggs under the surface of leaves. Those are all eggs packed on the undersurface. And these are bananas that have actually got a what's called brown spotted disease. So for a while, Orosema, or a Eucharidid that was laying its eggs into the actual bananas, was causing a major kerfuffle. And it's all sort of written up if you get Howard Evans' Life on a Little Known Planet. That's the only place where I have some decent information on what happened back then. But anyway, it went away, probably a DDT upset or something like that. Anyway, you get this nice little planidium, has various ways of getting down into the nest. It might be through direct attachment or being carried back by the ants themselves. Or we have a large group of wasps where they're laying their eggs and associating with immature thrips. Those are planidia attached on the thrips. And the idea is that ants are picking up the thrips and carrying them back as food to get back into the nest. They then get on to the ant larva over here, and here you got a couple of little planidia on a big Camponotus larvae over here. Then they always never develop on the larva. They wait until the ant pupates, and then they relocate themselves to the ventral part of the thorax of the ant. Usually there's only one, but in this case there's I think there's 4 on there. But that's a rare event. But the planidium, by definition, is something which is an external clinobionte, which basically changes through different life stages before it actually feeds on and develops hosts. And then they have a whole bunch of behaviors to get out of the nest, which I'll try and get to. So oviposition strategies, and these were summarized by more or less Clausen back in 1940, where we've got ovipositing into overwintering buds, into expanding leaf and flower buds, seed receptacles, which all I think are the same things. You can get incisions in leaf tissue at random on the leaf tissue, and the leaf tissue associated with thrips eggs, which I'll get to. And host associations of getting into the nest are first instars phoretic on the adult ant, or maybe on the insect prey of the adult ant. So that's where we're sort of leading off with different things. So story time. So we were at La Selva Biological Station, and one of Christiana Violau's students pointed out to me that there was some Eucharitids on a plant in the back of the station in the educational complex. This was Hemilia patens. And this Hemilia was basically something that was sort of a superstar back when Rob Colwell talked about this hummingbird that comes in and feeds on the flowers when they first open up in the morning, and they basically you've got these mites that are jumping off the hummingbird and into the flower and then back into the hummingbird, and it's sort of an interesting interaction. So here's my little wasp. This is that Ectotoma. This is the genus Capella salsifoces. And here's the ant sitting there on the Hemilia. And if you can barely see it, the ant is actually nectaring here. It's got its mandibles instead of a bunch of nectar, which I assume it's evaporating off to be able to go back and feed to the ant larvae. If you cut open one of the flowers, you see all these wonderful little planidia all lined up within the flower of a mature flower. If you look at the younger flower buds, you get these little clusters of white eggs, and those are basically the undeveloped eggs, and they're laid in masses of 20 to 40 by the Eucharitids, and they'll fill up these little undeveloped buds. When the flower matures, all these larvae start migrating up to the tip of the flower, and the idea is that as soon as the flower tip opens, those planidia start bailing out, and it's time to get out of that before the hummingbirds sort of come along in the morning. Here's some little planidia on the outside of the flower, and there's one just standing up there, just waiting here. And this is that nectar source. So now we have the flowers dropped, and there's just a bunch of nectar down there, which might be an attractant for the ants to come in. So this is my cheap ecological experiment in the field, where I basically categorize the sizes of the flowers and their growth, and I can see the immature eggs are only being laid into the young flowers. Mature eggs are being developing into those more maturing flowers. And then finally, the planidia is showing up just as those flowers get to be large. And then basically once they're out, then the planidia are outside of the flower. And that's when the hummingbirds are coming in and taking over on those particular nests. So again, we're ovipositing a young flower. You got these ovipositions of 20 eggs. You got larvae maturing as the flower develops. Larvae migrate to the tip, emerge through the minute opening as the flower begins to open. The hummingbirds cause chaos, and then the planidia scatter on the flower. And there's those nice little planidia embodied within the egg in there, and the other ones here are just sort of scattered around in the flower. Okay, so how do we get back down to the ant nest? And here's that ant in the flower, which is providing the nectar. If I open up the jaws of the ant and look at it and say, like, okay, there's the planidium inside that jaw of the ant. And here we have this 11 to 24 ants, planidia in their mouth parts, mostly in what's called the infrabuccal pocket, which is sort of a pocket. Ants don't really, it took me a long time for the ant people to explain this to me, but ants don't eat solid food. They take the solid food and they feed it back to their larvae. So what they're doing is they gather up solids into their infrabuccal pocket, they go back and they regurgitate it to the larvae, and then the larvae give them back nutrition in return. So this planidia has found a wonderful way of getting down into the ant nest by being associated with getting into this infrabuccal pocket. And then you can see that here's the host larva, here's a planidium sitting there, it's got located itself right on its throat, developing here. Those planidia start out from little tiny dart things, and then that's still the first instar. Those are the tergites of that when it started to grow and feed. As they develop, that first instar skin stays on there, which is really nice for me because I get to be able to slide mount that if I don't get the first instar planidium. And then we pupate within the nest, and in this case, we're pupating within the cocoon. So basically, we've got all of this nice little information, and we'll talk about semiochemicals for emergence in a minute, but there's, basically it's an easy way to get into the nest and develop on the host. This is some incredible work by Gabriela Perez and Jean-Paul Lachaud, which is basically looking at how the ants interact. How do you get out of the nest? And somebody brought up earlier about what about the trash pile? Well, here we have one that's basically grabbed on those wonderful long spines. It grabs onto those spines and uses them like little wheelbarrow handles to carry the insect out of the nest and just drop it onto the detritus pile. So the idea is that it sort of smells like an ant, but it doesn't quite smell like the right thing. So they don't want to kill it, but they don't want to have it hanging around. So it's the way of getting out of the actual ant nest. And it's not only that, but the ants will fight over these things. This is the one we're fighting. The ants will fight over how to get out of the nest. So basically, who gets to carry it out? Apparently, no wasp has ever been harmed in any of their experiments. It's just the wasp just sits there and waits to be carried out of the particular nest. And I've actually got images of where the ants will even be taking the cocoon out of the nest as the Eucharid is emerging from the nest. So I'll get back to that a little bit later. Eucharids, basically, this is the phylogeny by Murray et al. I'm going to use this as the sort of example of what we know about the particular groups. We have a nice little phylogeny of the group. We have fossil calibrations on here from Pterinomids, Perilampus, and Eucharids, all from Eocene amber deposits, which gives us an idea of when the actual origin of the particular group was back about 87 million years ago. So we get, and notice that this is, when you think about this, ant diversification is considered to be up to about 60 million years ago. So Eucharids really aren't evolving until after that period of time. And Elizabeth did some wonderful work that sort of showed that basically it's a lot of adaptive radiation of ants, of the parasitoids later on as they jump onto different ant host groups, such as ponerines, ectotomines, myrmecines. Now, these ones are on formicines over here, and more ancestrally, we have a group on myrmecines. So, introduction to the different subfamilies. Gallimielinae, this is something we know about. Clausen actually was the one that talked about these ones as being associated with a thrips egg, which is here. And they had these weird eggs standing up on end, which I thought, that's impossible. There's no eucharidid egg that could look like that. So I ended up going to Malaysia and Kuala Lumpur. And here I was basically had a revelation here of seeing all those little white dots on the fern leaves. And those are all eggs which are standing up on end. And the eucharid actually has a hook on the end of the egg. It hooks it into the plant, deposits the egg. And this is not a stalk. This is a secretion which is being dropped onto the egg on the top of it. And the idea is that that's probably what is attracting the Nylanderia to come along and collect those eggs. And here we have a larva over here with an internal planidium. So it burrows just underneath the skin to complete its development. So that's basically our Gallimielinae. We move up to Oracemines, which are all parasitic on myrmecine ants. And here's that internal planidium again, which is just underneath the larval skin. This is where we see the association with thrips eggs. This is a larva feeding on the, sort of like a teddy bear feeding on the child as it's developing. And these ones all develop, it's important here, they're all developing in naked ants. Basically, they do not have a cocoon. So the hosts don't have a cocoon in how they're developing. So they're just out there exposed within the actual brood pile. This is my daughter and I collecting in Argentina, looking at yellow trumpet flowers. And then this is what the eucharids can look like. And they're out there, they lay their eggs in Oracemini, the one subfamily. They have a specialized ovipositor that they use to insert eggs into the plant tissue. So all of those little punctures you see on there are egg punctures that are being laid by different females coming in and feeding on that. Here in California and Arizona, we have a wonderful little complex of things where this is Oracema simulatrix, which is basically ovipositing onto desert willow leaves. And it oviposits strictly around extrafloral nectaries on the leaves. And those larvae will actually migrate into those little swimming pools there to be picked up. And there's a planidium in relation to the ant. Our idea was always that the Phaedolae were coming up and feeding on the nectar and picking up the planidia and carrying it back to the brood pile. So far, we have not been able to find a single planidium within a Phaedolae worker collected on the plants. We have found them in about 20% of the Camponotus and Myrmecocystis that we've collected on the plants, ones that would more typically go after the nectar. So this led to a sort of a bizarre idea. Some of my undergrads actually discovered that they found some dead Camponotus in a Phaedolae nest. And the idea is, are the Phaedolae scavenging the Phaedolae, bringing it back to the nest to feed the larvae, and that's how the Planidia get into the nest. It's kind of odd, but this German undergraduate sat there and was working in Indonesia, and he came up with this. He was putting Planidia onto ants, and he says ants placed into the vials immediately showed an intensified but apparently inefficient cleaning behavior. After a short time, first locomotive dysfunctions were observed, followed by immobility connected with a typical twitch of the legs and antennae. In a final state, the ants did not show any vital signs at all. The Planidia are killing the ants. This is odd. I couldn't believe that. And also, unfortunately, never ran any controls. Something advice to everybody to follow up on. And I'm going to come back again to that one. So Oracema and its host ant, how does it deal with its nest of being in the open in the brood piles? The nice work by Vandermeer in 1989 showed they have, when they're in the nest, they have exactly the same cuticular hydrocarbon profile as their host ant, which is in this case a Solenopsis. Then we're coming down here. As soon as we get outside the nest, we have a differentiation, and we have a differentiation of the adult ants as well. But they have this critical sort of recognition factor while they're in the nest. And again, they'll sit there and they'll be in the nest, and once they emerge, they probably got about 3 or 4 days, which I observed in Texas, before the ants recognize them and start ripping them apart. So you smell like your host. Oracemines are kind of a cute little group in that they, this is some work by Austin Baker, my grad student, and basically we know that ants, the Phaedolae, based on ant research, the Phaedolae came across from the New World to the Old World about 27 million years ago. Oracema actually went the opposite direction and colonized as a single monophyletic group that came across about the same period. So basically they're utilizing the host and going for a backwards invasion. So that's what leads to the diversity we see now. Less about that. Eukaryotic, we're looking at now at this group where we have a single evolutionary adaptation to attacking hosts with a cocoon. They have similar behavior. There's a larva feeding on the host, but it's external now. It's not internal. There's one genus that's actually internal in the larval stage. But they, again, laying into flowers, and we can get development occurring within the host cocoon. We get Psilocaratini at the base here. These are ones that we actually have something which is comparable in Baltic amber to this particular group, and this is the one that I'm working on in South Africa that I'm trying to find the ant host for, because we really don't know what it is for that one particular group. In the other Psilocaratine lineage, it's a hyperponera, a small ponerine ant that we're after, so we know something. But we need this one critical because it's probably the sister group of the rest of the eukaryotians. We look at the Eucharitini. Within that group, we have basically one lineage which is attacking all the formicine ants. So these are basically going after Camponotus. They're laying their eggs into buds in large masses or into fruit. And they seem to be utilizing the frugivorous habit of the Camponotus, and which are basically, they're coming up and they're collecting the Planidia and taking it back into the nest chamber. We have another Eucharitini, which are all attacking Ponerines, Ectotomines, and Myrmecines. And again, these are highly active Planidia. In this case, they're very active. And here's a single Capella individual that's picked up all these Planidia that have attached onto the ventral side of its body. So basically, they're coming across and attacking the ants that way. So we're sitting there. We got this one idea of coming up here. Okay, so we have Ectotoma rubidum. Basically, this is one thing I wanted to talk about is how they get out of the nest. There's all this interaction that's going on here. When you compare this with what's going on in the Oracemines, again, work by Perez-Lachaud and all, showed that they have cuticular hydrocarbons which are they're similar, but not the same as their host. So their host Ectotoma has a broad range of cuticular hydrocarbons. This is the genus Dilacantha, which occurs in the same nest. It has a different set. And we have Isomorella, which is another set, has a different set of hydrocarbons. So the idea is that they may actually be mimicking compounds, which is why we see this direct reaction of the ants reaching out to them as soon as they're starting to emerge as adults and trying to get them out of the nest. They smell somewhat like their host, but not enough that they want to do it. And the idea of whether they're producing them themselves or not could still use some good chemical ecology. A little bit of, as we sort of wind down, I want to sort of drift into some of the bizarre Eucharitids that you can come across. So this is Diacylothorax males, and basically they have these long flattened spines that come out over the gaster. My hypothesis is that spines are actually there to protect the wings. When you see how the ants grab onto them by those spines, they may be a little bit more protective. This is Galliera. It looks like little lima beans. Basically, basically the gaster can tuck up in there, and they're obviously being protective in some way. Look at their habitats. This is Cabeza de Buey in Argentina. We get basically the ants down there. This is Javier Torrens, my colleague down there, and he's basically digging up the ants. And you can see these are there's where the planidia have attached to the host in those cases. Just these little larvae just sit there and they'll just wait for the ant host to pupate. This is another one, Ectotoma. Over here on these plants, those are all planidia. They're all just, again, there's an idea of how are they getting down into the nest. Lots of studies could be done to sort of follow up on that. But do they rain down on them? Are they doing something else? So conclusion of the whole talk. So we talked about transcriptomes and what we're doing there for the higher calcid relationships. We came up with a combined AHE and UCE analysis that sort of put all this stuff together. We talked about atomization of the calcids, where we now are up to 50 different families of Chalcidoidea, mostly coming out of Terramality, which is basically now gone from 35 subfamilies to only 4, I believe. You got the Eucharitid phylogeny that we're looking at, and there's some actually conflicts there we get with the Planidioclade phylogeny over here, but I didn't talk as much about that. We have Oracemine phylogeny, which shows these big invasions, which are concordant with some of the ant histories that are going on with some of these groups and how these things moved around the world. And lastly, I wanted to sort of rhyme off with, yes, Eucharids are important and people do care about them. Recently, I came up with Val Rogers, actually was the first person on Facebook, he posted a picture of a Oracema minutissima from Hawaii, which it's not supposed to be there. Turns out that it's somehow been accidentally introduced. It's a parasitoid of a little red fire ant, and it's going like gangbusters all around the island. I've got a grad student, Ashley Blee, who's now working on trying to find out how, what is the pattern of origination of this parasitoid going against this Wasmannia, and also whether it's going to have some potential for control of the fire ant, which it seems to have some really good promise based on the numbers that we're finding in Hawaii. So systematics is a marriage of the laboratory and the field. I've enjoyed a lifetime of going out and collecting all this biological information and also bringing things back to the lab and putting them to good use. It's also been a fantastic journey with a whole bunch of grad students and other colleagues and things like that. Without them, this probably would not have all come together in any realistic way. And I really appreciate everything that everybody's done on this. And my little plug for the Universal Chalcidoidea database over there, if you want to go out and find out how to search information on taxonomy and biology and host relationships of calcids, please go there. And with that, this is the name list of people that I would always like to appreciate and call them helpful in all these different projects. And with that, I will stop.
Well, thanks, John. That was an incredible presentation. Hey, there's John right there. Look at that. He's in the car.
I am there. Welcome from Newport, Oregon.
Awesome.
We're almost to Waldport, but not quite. It's
So we've got
raining.
a bunch of people here. We've just finished your lecture. I think this would be the time for people to ask any questions that might have questions for you. Oh, we got some people clapping at the moment.
Very strange to watch yourself giving your presentation for a couple of minutes.
I can ask a question. I always like asking questions of John. I did it for a really long time. So something that always bothers me about Eucharitids is their seeming lack of interest in dolichoterine ants. And I ask this question of John maybe once a year to see if there's been any updates. Slash, I think other people might find it interesting. What's going on with the dolichoterines, John? Why do the Eucharitids seem so disinterested?
I have the same answer for you every time you ask that, which is I do not know.
I just hope one day we find out. It's so bizarre.
I mean, I think the other thing that weird is that when you think about the fungus growing ants, you've got all these Eucharitids in the tropics, they're all laying eggs into vegetation. Why has nothing ever made it into the fungus growing ants, which are collecting those leaves and bringing them back direct? And I've talked to Ted Schultz and other people about this over the years. And one of the ideas is that fungus growing ants are actually very meticulous at picking out bad spores that are coming in on those leaves. So they can probably recognize the planidia as something being evil, whereas everybody else doesn't have it in their search image, so it doesn't get in. And also fungus growing ants often cover their larvae with all sorts of mycelial fungi and things like that that may also stop them from ever getting to burrow into the ant or something like that. But yeah, there's actually, it's a fairly restricted group of ants that they're attacking. But dolichoderines and the atine ants are just not in the mix.
Just a quick follow-up on that. I know that there are some diapriid wasps associated with the attines. Are they not actually parasitizing the attines? Are they like hanging out in the flies in like the trash heap or something?
I think they don't really know what those diaprids are doing. There's always this thing about, are they actually attacking the flies that are also potentially within the nest? So they may be attacking something else. They may not be attacking the ant brood or anything like that. So I think there's largely an unknown on that. But I know that they were looking for dolichoderine parasites in Argentina, and they were never quite certain whether they were actually going after the ants or the flies that were resident within the nest.
Thanks, John.
Just a little aside, I think I gave one of the when I was at in Washington as a postdoc, I was associated with giving one of the big plenary speaker things out at the Beltsville auditorium, and we had David Hull, the philosopher, give a talk. And I remember over dinner, we asked him, "Well, what kind of slide, would you need? Do you need a overhead project? Do you need a slide projector?" And he says, "No, no, we don't need I don't need a slide projector." "Oh, so you need an overhead?" And he goes, "No, no, no, I don't need an overhead." "So what do you need?" "Well, I just need a light on the podium so I can read my talk." And we're all like horrified, going like, "Oh my God, he's going to read a talk to 500 people." And we're going like, "No." And it turned out he was the most eloquent reader that I've ever, the way he chose his words was absolutely amazing. So it was actually a great, great talk. But man, it scared us. At least I have slides. I don't have as eloquent things to say, but I have slides.
And movies.
And movies, yeah. And Eucharitids because they're cute.
They're all right.
So I have to say, as part of my adventure, I've never given a Zoom from a car, and also the car rental we have is a big giant red Jeep. It's actually been exciting to drive around here, and there are no Eucharitids out at this time of year here.
John, thank you very much for your generosity, allowing us to use your talk and joining us from the car. Do we have any other questions? I don't.
Curious. Hi, John. Nice to see you. Chalcidoids are such a big group, and I've noticed that a lot of your work involves a lot of collaboration and just kind of speaking about how some of that kind of went on and some of the perhaps even advice that you can offer regarding some of that, where um, you do your specialty, but then you also rely on some of the aspects from some of the other people. I mean, that's a lot of work that you've done over your lifetime, which is impressive. So I'm just kind of curious about that, as much as what you can offer.
Well, I think a lot of the large ones, like the large, the studies we've done of the whole super family over time, and which ends up with 55 collaborators helped to write this book. It's dealing with a lot of people, and the Chalcidoid community is just a really friendly group of people. I've traveled around the world. I've met with a lot of them. We get to be friends, and so that makes it a little bit easier to get people together to do something like that. And we had a initial workshop back in 2005 that was part of an NSF where I brought everybody to Riverside, and we had a whole 2, 3 days of presentations to go over and talk about everything we were going to do. Eventually, in 2011, we brought a whole bunch of people in to do morphological character coding. So all had to come to Riverside and hang out and do coding and things like that. So cook dinners and have fun and everything else. And then finally in 2018, as part of the NSF bigger project, we had another workshop in Prague and getting everybody together and spending a couple of days of going over characters and talking about relationships and working out, say, okay, so how many families of Chalcidoids are we actually going to have? And making sure everybody had their voice and what they wanted to say. And some people were adamant that they weren't going to change their ideas of the relationships within a group. So we had to go with that. And other people were saying, okay, let's fragment this thing and move on and however we do. But the day we ended that workshop in Prague, everybody was happy that they'd had their chance to participate. Then it was a case of, 2018, we finally had our final trees coming together and then trying to get everybody to write the chapters that would go ahead with those things. And that takes a lot of cajoling and following up on people and helping and, getting them to write their chapters and get it all together. And I know, Lourdes, you're going through all that with the Coleopter of Canada book and whatever. And, it's just I think it always, and there were a couple of times when it got really dicey and we thought the whole thing was going to fall apart, and because there was a couple of critical people that we need to include, but just a little bit of keeping everybody happy really worked in the end. And then I destroyed my knee in Romania, and I had three months of basically sitting on the couch and doing nothing, which was a great time to edit all the chapters and put all that together.
So what about your Entomologics? I mean, what is it about those? They're just, you've never been able to include them in any kind of analysis because they're just so rare.
No, they're in the analysis, but they're bouncers. They just won't go into another group. And then every time you run a different analysis, they come out in a slightly different place. So there's no certainty about where they are. So until we get more material, maybe try new methods or something like that, they're not going to have a home that's there. So it's, that's where we're left with some of those groups. And some of them we just don't have enough genetic information for too. Like there's one genus, Jembia, which is from Israel and Yemen, extremely rare. We have sequence for it that we got from, but only just some, I think 13 AHE genes that we were able to get out of the, 500 that we were doing. It comes out in the right place in the planidioclade, but in the whole Chalcid phylogeny, it's just sort of there's not enough genes, and it might be one of the reasons it doesn't come out so nicely within that group. But in Cetus, we just got to hope we get more specimens. And I think Jean-Yves is in the process now of trying to sequence every single Chalcid genus for at least 2 taxa with UCEs. So we'll see what he comes up with out of that particular result. That was the other big help in all this too, was having Jean-Yves and Astrid Cruot over in France. And they were basically doing a complementary molecular data set for UCEs while we were working on AHEs. So actually putting them all to, getting the idea together that we could put them all together into a single analysis was a big thing. And it actually, one or the other was not going to resolve the whole thing, but together I think we came up with a better result other than for these inserted seeded taxes.
Thank you.
We can never be finished with something, right?
Yeah, I would love to have you back in person when there is news about it.
Someday.
So where can people get your book?
Where what? Say? Oh, they can buy that from Cabby. Let's look up Chalcidoidea of the World. It's a great table talk book. It's got tons of images of SEMs and color images and everything else like that. And I think it's worth even a beetle person wants to get it.
Absolutely. I mean, you said it around people.
There it is. Luke showing his little copy right there.
Look at that. That's fantastic.
We have a few people saying thank you in chat for the superb presentation.
Thank you.
I'm mostly a fly person, but they were absolutely gorgeous. I loved the photography.
Well, I've been working 45 years of my life on these things, so they must have done something. Hi,
Yeah,
Warren
thank
and
you
Joel.
very much.
I guess say hello to Warren and Joel. I lived with them for like a year here in Washington.
Thank you.
It all started in 1990 in Charlotte.
It was great fun.
Well, thank you so much.
Good to see you. Great talk, John.
Okay, thank you.
We'll get your book.
Enjoy the meeting.
Yeah, say hi to everyone at ESA for us.
Yeah, I
We
wish we
will.
could be there.
It'll be a lonely place.
They look Christian.
Oh yeah, sorry. I should introduce the other car partner there.
Hi, Christiana.
Working away on my talk.
You want another talk? We can have one right now.
No, I'm not ready.
Go on, next one.
That's hilarious.
Okay, on to Waldport.
Fantastic.
Thank you.
Okay, bye.
Thanks, John.
Thank you.
Bye.
Bye-bye.
And also, I'd like to thank everyone for joining us virtually and staying engaged while we wait for normal museum operations. Thank you all very much. It was actually very fun. I think we did a good meeting. I was a little bit worried about it. So I think Alan has his hands up. Alan, do you wanna?
Just a quick note to Gary. I think he keeps track of how many attendees are. We had 38 participants.
Amazing. Thank you all.
Thank you.
Okay, so if that's all, can I have a motion to adjourn the 1255th regular meeting?
So moved.
Second.
Do I have a second? Thank you very much. It's moved. Thank you all for being here. Good night, and I hope to see many of you in person soon.
Have a good night.
Good night, everyone.
Good night.
Good night.